here.
Thanks. Thanks a lot, Brian. Does the microphone work? Okay, good. Thanks for coming, and thanks for inviting me. So, as the title says, I'm going to be talking about systems biology approach to understanding the immune response to tuberculosis. So, what is the systems? The, oh, you can do it. Okay. Okay. So first of all, I want to start with a brief definition of what the system biology approach means to me and to us, in the context of TB, but more in general. And so what we try to do with the systems biology approach is to integrate and synthesize data that are coming from several model systems, and that could be the human system, an animal system, bacterial system, and computational models. So integrate all this source of information and try to use them to explore complex biological systems. And at the same time, try to address and hopefully answer some of the open and relevant questions. One of the goals, and that's my goal, is to build some mathematical and computational platform that, first of all, reflect the human biology, and they can give us an opportunity to understand some basic principle governing these systems, and then generate testable hypotheses or perform experiments with the mathematical model, a computational model that cannot be performed in vitro or in vivo. So those are, very general. This is a general definition and why we do it. Now, how we do it? Let's say the main idea and methodology is to try to go from a reductionist approach to a reconstructionist approach. So we have control in vitro experiments and try to use this information to build the intact system in vivo. Now, it sounds easy, but where do we start in doing this? Here's where I try to, and I use a statement by the Nobel laureate Sidney Brenner, which is elegantly described in this book by Dennis Noble. He gave a talk a few years ago here. So this statement is very comforting because he said the analysis must start somewhere. It doesn't matter where, at any scale or organ, at any level, I mean, whatever you're comfortable with. And in the best of all systems biological world, we will eventually meet up anyway. So that means that this speaks to the true nature of systems biology, which is interdisciplinary. It's definitely a teamwork, so we can have a molecular biologist to collaborate with an epidemiologist, and it's a different scale, a different study, different data, but they address the same issue, and they try to build a model or a computational platform that try to describe the whole system. And so eventually they will meet up and they will formulate link of these different scales. And that's the overall idea. Now, I'm going to give you two examples. Most of the talk is about tuberculosis, which I call theory-driven modeling. So we don't have a lot of data in TB because the animal models are not very similar to the human pathology, human disease. And we cannot really get longitudinal data on the same subject. But we do know a lot about the immunobiology and the epidemiology, so we can build into the model this knowledge of what are the main players in terms of cell types, what do they do, and what do they need to do in order to mount a protective response to the pathogen. So I will walk you through some of these facts and how we approach this complex system with computation and mathematical models. The last part of the talk is about new exciting work that I'm doing with Pat Schloss in microbiology and immunology. It's about microbiome. And in this project, we are basically at the other extreme, where we have a lot of data, longitudinal data, and we need to make sense out of it. So it's about mainly network inference and devising some dynamic modeling platform to describe the different changes of microbial communities over time. So let's talk about TB. It's a deadly infectious disease which is caused by Mycobacterium tuberculosis. In the majority of the cases, once exposed, the host progresses to what is called latent infection, which is asymptomatic. It is not infectious, and it can last for the entire lifespan of the host. From 5 to 10% of the exposed experience what's called primary TB, or active TB, which is infectious, and if not treated, can cause death of the host. There is a possibility of clearing the infection upon exposure, but that's not really clear. The problem is, as Brian mentioned in the introduction, that we have a pool of latent infected individuals, which is pretty large. We're talking about 1/3 of the world population. And what happens is, during the lifetime, there is a chance, which is quantified approximately 10% during the lifetime of the host, chance of reactivation due to many different causes. But the result of that is that some of this latently infected individual experience active TB. And so we have a lot of latently infected cases that need to be treated in some way, otherwise they're going to reactivate. And just to give you some numbers, we have 9 million new cases per year. And I don't know, what is it, 5 minutes since I started the talk, probably 15 people already died of TB during this talk, which is not encouraging. So let's talk about the course of infection. Very general. I mean, it's really ABC of immunology. But what happens upon infection in the lung after the bug is inhaled in droplet, and it basically reaches the bronchi and the alveoli in the lung, it triggers a cascade of danger signals and cytokines and chemokines that track different cells to the site. Most of them belong to the innate immune response. But some of them, like macrophages and dendritic cell, they play a major role in the immunobiology of TB. Macrophages are basically the preferential host for bacterial proliferation. And most of the macrophages become infected, and they give an opportunity to the bug to proliferate intracellularly. And some of them progress and they burst because they cannot contain all the bugs inside. Upon infection, they start to produce several chemokine and cytokine. The main one is tumor necrosis factor, which is very important. Some other cells, like dendritic cells, they do become infected, but they change phenotype. They become migratory and mature into what they call antigen-presenting cell. What does that mean? It means that they start to migrate through the draining lymph node and instruct another class of cells called the lymphocyte, and they tell them about the enemy, basically, which is the bug. They do that by presenting pieces of the antigen, pieces of the bug on the surface, and that particular part of the bug has to match a receptor on the lymphocyte on the T cells. Once this match occurs, the T cells are called primed. It means they are instructed and they know what the target is going to be at the site of infection. They start to proliferate and become effector T cells. That happens either in the lymph node or on the way to the lymph node. So once they become effector, they express different receptor and ligand, and which allows them to get out of the lymph node into the blood and then reach again the site of infection. This process is basically the mounting, it's called mounting of adaptive immunity. And it takes time, obviously. It doesn't happen immediately. So in the meanwhile, I mean, the infection is going on in the lung. And what happens is all these cells, they co-localize and they surround the core of infection that is going on for the first couple of weeks in the lung. And they form this spherical collection of immune cells called granuloma, which is the hallmark of TB infection. And so the granuloma, in a way, physically constrain and immunologically restrain the bacterial growth. It's beneficial to the host, but also to the bug. And we don't know that much about how it forms and all the mechanism of development and maintenance. What we do know is that the T cells, the lymphocytes that are coming back to the site of infection, they do very important things. One of which is the macrophage activation, which is basically a necessary but not sufficient step for protection. And when a macrophage is activated, it can kill the intracellular bacterial load. It can kill also the other bugs. So it's very important in mounting in the protective immunity. But they do also other things, such as killing infected macrophages directly, these T cells, and also indirectly through apoptosis, which is a programmed cell death, where the infected macrophages dies. And all this process basically release some of the bug intracellular, and they continue this process, the continuous turnover of macrophage infections. So the granuloma is basically a very dynamic entity, and there is a continuous turnover of cells. It seems pretty stable and pretty, it seems like not too many things are happening if you look at an X-ray, but there's a lot going on. So what we want to do is try to look and focus on the granuloma. I mean, at least in what I'm going to show you in this talk. And then typically what happens is those are granulomas in monkeys. So those are real granulomas. Typical structure is a core, as I said, of infected macrophages and the bug, the bacteria, extracellular bacteria. Then there is another layer of macrophages that can be activated, and there is the outer layer, a lymphocyte cuff, which basically comprises mostly of the effector cells that I was talking about. They reach the site and they activate macrophages and they contain the infection. This process has to keep going. If something is disrupted, what happens is there is an inadequate control of the replication, and the bacteria can escape the granuloma and determine what's called dissemination. And that results in secondary lesion, either in the lung or most often in other organs. Generally, the replication is controlled, and as I said, most of the people experience lethal infection. And the granuloma becomes stable, stabilizes, and undergoes a healing process, which involves some fibrosis. In general, all this process does leave the lung with a little bit compromised. So there is no complete recovery from TB. This step is accelerated by antibiotic treatment. So even if there is a treatment, after you get treated and you eliminate TB, somehow your lungs are still compromised, so there are chances of relapse. Now, again, from latent TB, there are chances of reactivation due to, most of the time, it's immunosuppression. You can think of HIV patients, the top cause of death of HIV patient is tuberculosis, because basically the immune system goes down and the TB takes over. But it could be due to age, for example, or another infection. So that's the course of infection. Do we have enough data? We do have data, but not as many as we wish. That's always the case. There are several established animal models. So there is the non-human primate, the zebrafish, mouse model, guinea pigs, rabbits. The most used and popular is the mouse model, which has a lot of immunologic reagents. So, and it's been studied a lot. There are several limitations, though. It cannot reproduce the typical course of infection in humans. So, for example, in a mouse, we have chronic infection, and it's progressive. So sooner or later the mouse is going to die. And it reaches the peak of infection pretty fast. So it's very unique. And also the pathology is different. The granulomas are not exactly the same. Then, so the best one is non-human primate, which is very similar in many aspects of the TB, stages. The problem is that it's very expensive, and there are year-long experiments, and there are very relatively few reagents available. And so this is the best place to develop a computational model where we can supplement and complement the research done in an experimental lab with some of the mathematical model simulation. And that's what we did. So starting 10 years ago, I started to work on modeling temporal dynamics of cell bacteria and cytokines in the lung. We developed some ordinary differential model of TB infection. And so one of this work, for example, addressed the concept of reactivation threshold, and we use the model also to do what I call virtual clinical trial. So in this particular work, we look at how neutralizing TNF can perturb and basically disrupt the granuloma, which is actually what happens in many of these anti-TNF treatment that are advertised on TV too. So many of these commercial, they warn about don't use Enbrel. This is one of the most famous ones. If you have been diagnosed with TB, because that's going to cause reactivation, and you cannot start that. You cannot start a treatment unless you do some full course of antibiotic for TB. But we do not know how much TNF we need in order to contain the infection. So there could be a balance of, well, you can undergo the treatment, but you cannot. There's going to be a reactivation threshold for the TNF that is bioavailable at the site. That cannot be done experimentally. So we use the model to kind of make some prediction on that, which were confirmed later on in some experimental studies. But also we use the model, to perform virtual clinical trials and try to mimic the different mechanism of action of drugs and see which one was causing this reactivation of TB. We also look at two compartmental models where we look, as I show you at the beginning of the cartoon, where we have the lung and the lymph node, and model trafficking of cells back and forth. The one I'm going to talk about basically merges all this into what we call a granuloma hybrid model, where we have spatial temporal dynamics in the lung and some ODE in the lymph node. Now, another couple of slides on the methodology, and then I'm going to go into some of the results. But the model that I'm describing are fairly complex. They're large in size. They have a lot of parameters. Many of those parameters are unknown, or at least we are very uncertain on what the value is or should be. So how do we deal with that? We use a very powerful methodology, which is uncertainty and sensitivity analysis. It's in two steps. The first step is uncertainty analysis and uses Monte Carlo methods and Latin hypercube sampling, which I will illustrate later on in the next slide, to explore the parameter space and look at what we can actually get by varying this parameter in these ranges, what type of output the model produces. After the output are generated, the thing that we want to do is try to quantify how the variation in the output that we're looking at. An output could be the total number of bacteria. How we can apportion this to different biological mechanisms. And we use generalized correlation coefficients or variance decomposition methods for that. I'm going to show you this particular coefficient called partial rank correlation coefficient. The key feature of this sensitivity indexes is that they can quantify relationship over time. So certain mechanism might be relevant early on during the infection and not important later on. For example, if you look at viral dynamics, we want to look at the first week of infection rather than 200 days post-infection, which would be the time frame of TB, for example. We publish a highly cited review in JTB, which is specifically targeted to performing this analysis globally in the parameter space. So briefly, what it is we define a mathematical model here for I'm showing a differential equation system. The state variable x and parameter theta, and the output is a function of these two. Then again, for ease of illustration, a parameter vector that has three parameters that we don't know about. Well, in the Bayesian approach, what we do, we specify some a priori distribution for these three parameters, depending on the knowledge we have on the system, on the data, on the literature. And if we don't know that much, the best choice is a uniform distribution. We need to have a reasonable guess of the minimum and the maximum values of this parameter. But that's an assumption that is pretty neutral using a uniform distribution. If we know a little bit more about the more frequent behavior, well, we can use a normal distribution or another distribution that is biased. But that's the approach. Defining a priori distribution, then the method is stratified sampling without replacement, which means once we define a sample size, here is 5, just for ease of illustration again, we divide the interval in 5, in this case 5 equiprobable intervals, sub-intervals. And so when we start to sample, we pick a value in this sub-interval, for example. The first one is actually this one, A1. When we pick a value here, this sub-interval is going to be eliminated from the sampling scheme, and so on. And so once we define all these values for A, the parameter A, we can go on and do the next one for the parameter B and C. So it's the main hypothesis is that the sampling and those parameters are independent from each other. So then the next step is defining the parameter matrix and plug in this parameter into the model and get some output. Then we can correlate simply the raw value that we have here, and this will be the correlation, Pearson correlation coefficient. Or we can rank these values and use a Spearman correlation that will kind of filter out a little bit of nonlinearity. Or we can use the partial rank correlation coefficient, which is the best choice, and takes into account basically some collinearity between all this parameter and the output too. The LHS is very is great because allows an unbiased estimate of the average output, and but with much less samples. So we can, for example, have the accuracy of 10,000 samples in the simple random sampling scheme. We can achieve the same accuracy with 1 log less samples, which is particularly important if the model takes a long time to solve. Now, having said that, let's go back to TB. So those are the tools that we use routinely to basically address the modeling, building process, validation, and post optimization. Now, I show you this cartoon where we do have granulomas in the line, and we have the lymph node that generates effector T cells. And so the reasonable idea was to track granuloma formation in the lung with an agent-based model, and track the T cell priming and the generation of effector T cell in the lymph node with a temporal based model, or an ordinary differential equation. So you probably know, I'm not sure if you know what an agent-based model is, but very fast. Basically, this is the agent-based model running, and it comprises 4 components. We need to define an environment. So what is this virtual square is? And we need to populate the environment with agents and some variables that can represent some diffusion molecules. And we need to define the interaction between the agent and the environment, between the agent and this continuous variable, and also the time scale for this interaction. The whole code, the whole program, the whole platform is usually implemented in either in C++ or in an object-oriented programming language. But there are also some software available like NetLogo, which are not as powerful though. So NetLogo? Oh, okay, okay. So okay, yeah. Well, so specifically to TB, what we have it's 2 millimeter by 2 millimeter. This is the environment that we use, so we need to kind of give some content to this grid, basically. And it represents a 2 millimeter by 2 millimeter section of the lung parenchyma where the granuloma forms. It is divided in microcompartment size to the diameter of a macrophage, which is 20 micrometer. So based on that and the average speed of cells, we define also the iteration and the movement of this cell. So basically every 10 minutes, we update movement and other changes of state. And the grid, we have agents that are macrophages. The color here means a different stage. We are resting, infected, chronically infected, activated. We have continuous variables that are bacteria that don't move. They are constrained within a single microcompartment. But we have some other continuous variables such as tumor necrosis factor, as I said, or chemokine that diffuse throughout the grid. We also have other cell types like T cells. They can be two in a single microcompartment. They are smaller. And we place randomly on the grid vascular sources. And those are the sources, if you see the gray spot here, those are where the cells are coming in, macrophages and T cells. So just to give you an example, I mean, there's so many rules and knowledge that is embedded into the model. But for example, a resting macrophage can become infected once it uptakes a bacteria. Then only these two types of macrophages can become activated. If they become activated, if they're infected, they can kill the intracellular bacterial load, or they can also kill the extracellular bacteria. Once there is a certain threshold, they cannot be activated anymore. And the fate of this chronically infected macrophage is to die, either by bursting or killed by other cells. Those are just some of the rules, but there are many, many, many to list. Anyway, what we did with the model was to try to replicate some baseline infectious scenario. And we look at 200 days post-infection, which is easy to do with the model. I mean, there's no time constraint. We can run it. This model is pretty fast. It takes approximately 30 minutes to run. So remember the Latin hypercube sampling or the uncertainty analysis. We can run several runs of this to explore different mechanisms and the ranges for these parameters. The idea is to replicate some known scenario like containment or clearance. And here in this plot, you can see the time on the x-axis and the y-axis, the total bacterial load. We try to replicate here 4 different scenarios that are described in snapshot here at day 200. So as you can see, the clearance scenario, it goes back to the original initial conditions. So it clears the infection. And then in the containment, we get a granuloma that is fairly stable in terms of size and in terms of total bacterial load. And those are two situations that we can call uncontrolled growth of the bacteria. And we can get those by knocking out, eliminating from the model mechanisms that are related to tumor necrosis factor or the interferon gamma. So mainly this represents an immune system that cannot activate macrophages, and this is what we get. And this one is more complex because the tumor necrosis factor can have an impact on many, many mechanisms. Now, we did use uncertainty and sensitivity analysis to target specific mechanisms, which are parameters in the model, and to try to identify some factors that distinguish between different granuloma outcomes. We were able to identify host, elements, and bacterial factors. I'm going to list just some of them, just to give you an idea. The degradation and diffusion and secretion of specific chemokine and tumor necrosis factor is very important. The arrival time, location, and number of T cells within the granuloma is key, as well as the movement and the recruitment, especially for a certain type of lymphocyte. And of course, macrophage activation rates. If we have more activated macrophages, we can definitely kill more bug and probably resolve the infection. Also, on the bacterial side, we target intracellular bacterial growth, and that is definitely a key factor in disease development. we found out that the slow growth facilitates survival. So it's kind of an escape mechanism for the bug to not grow very fast, which is a unique feature of TB. Doubling time is 24, 48 hours, which is very atypical. I mean, viral infection, they double, and even some bacterial infection, they can double in a matter of minutes. So the slow grow makes at least the initial part of the infection undetected from the immune system. And that's probably important to establish that niche that allows, the formation of the granuloma and the persistent later on. And overall, we found that bacterial numbers are a strong contributing factor to the size and the structure of the granuloma. Now, remember the branch out approach that I described at the beginning. We must start somewhere, then we can, either zoom in or zoom out and go somewhere else, a different scale, a different organ, a different level. We could target TNF specifically in macrophage activation and kind of zoom in a multi-scale fashion. And in one of these green pixel that we're moving, we can also embed a differential equation system that describes some signaling, intracellular signaling, which we have done it. I'm not going to talk about that, but or we can look at bacteria and try to model some metabolic transition into the bacteria life cycle. Right now, they all have a fixed proliferation rate, and so they don't really react and interact with the environment. But the thing I'm going to talk about today is to branch out and try to model more mechanistically T-cell priming and the generation of effector T-cells and recruitment, especially with the purpose of suggesting vaccination or immunotherapy strategies. How did we go about it? So all the simulation that you saw in the background so far, they assume that there was a certain time point after infection where T cells, which are those purplish cells that you can see coming in from the vascular sources, they come in after a certain time. And that assumes that after that time on, we have an unlimited pool of effector cells that can come in. It's like a perfect priming that it always takes place, which is not always the case. It's never the case. But that was a reasonable assumption and simplification. So the recruitment by then was basically stochastic. What we tried to do was, okay, well, but if those mechanisms are important, why don't we try to model them in a more deterministic, mechanistic way and try to build a virtual model of the lymph node compartment that takes information from the agent-based model and returns some fluxes of cells and generates some fluxes of cells that can be input onto the grid. So there is like linking mechanism between two different modeling platforms, if you like, agent-based model and an ODE, but also try to go back and forth between discrete and continuous variables, because this is going to be an ordinary differential equation that generates fluxes. Anyway, we use, and this has been published, but we use the experience that, on other models that we published before, where we had a lymph node compartment that was an ordinary differential equation system. And so we simplify some of those equations and build a lymph node ODE compartment with 9 differential equations that represent naive CD4 and CD8, prime T cell, effector CD8 and CD4. So the infection in the lung drives basically T cell priming through antigen presented cell migration. The efficiency of priming is also important, and it's a parameter of the model. Those fluxes of effector T cell, then they're kind of giving us the number of cells that can migrate and can possibly be recruited to the lung. Those are some of the equation. I just want to show you, the way we built it. Basically, there is a lot of theory-driven modeling here where we know that those are antigen-presenting cells coming from the lung, and they do interact with naive CD4 to generate some precursor T helper cells, which proliferate, and then they become effector cells. So this is the component, and some of them, they migrate out, those components, and then they reach the lung. So those are the equations that we track. In similar fashion, we have the CD8. More specifically, again, those are the naive CD4 dynamics. These are the effector CD4 dynamic. And those are the same effector CD4, but in the lung. We can look at specific mechanism, which is this parameter, K14 is basically the priming efficiency. It's a likelihood basically that if you have an antigen-presenting cell and a CD4, what's the probability that that interaction generates an effective T cell? So we can manipulate that, at least in the model. It's harder to do it experimentally, but that's part of the systems biology approach, right? So another thing we can do, we can try to see, well, what if we have more antigen-presenting cell migrating to the lymph node, which means more dendritic cell they are able to migrate and do some priming. It's not about here. It's not about the quality of the priming. It's about the quantity. So this is just one example of what we could suggest as testable hypothesis and possibly. I mean, this is a little complicated right now to design, but it can be done. Anyway, the idea here was, okay, we have a containment scenario. Think about those 2 billion people in latent TB. So how we can eliminate and treat or cure the infection and basically resolve this granuloma? Well, here we target 3 different mechanisms. One is that parameter I showed you, the CD4 priming. Here we try to enhance both CD4 and CD8 priming. Here we just enhance the trafficking of antigen-presenting cell. Well, this mechanism, the CD4 priming and the antigen-presenting cell trafficking, they're very efficient, and at the end, they can resolve the infection, so we can actually create the granuloma. But this one doesn't work. We speculated on why it might not work, but some of the experimental data showed that CD8 effector, cytotoxic activity is very important early during infection rather than late. So if we actually do this at the onset of infection, we always get clearance. So the CD8 priming is negative impact on granuloma formation, if you like. On granuloma dissolution. And so this is one particular application where we can tune some parameters in the lymph node and see the impact on a different scale, on a different organ with the model. Now, experimentally, this will be complicated, but there are data showing, for example, some dendritic cells that are pre-primed and they are very, very immunogenic. They can give similar results. It doesn't be shown in TB, but and also at the same time, T cells that have different different T cell receptor, and so they basically belong to the category of enhanced priming, so they're more easily activated. It's hard to devise it again, and remember that we don't have data over time on single subject in TB because this happened in the lung, so it's tissue and it's not blood, so we cannot get samples over time. Okay, so I want to talk now. I don't know how much I have left, but the last 5 minutes or 10 minutes on a completely different research project, which is data-driven, if you like, and it's about microbiome in the gut, microflora in the gut of mice gut. So it's a system biology approach to do what? To identify, quantify, and predict the overall structure and architecture of the microbial communities in mice under different conditions. So, and the idea and the goal is to infer the network that regulates this dynamics and try to do it with a data-driven dynamic model generation, fitting, and selection algorithm. It's something that I worked on in the past on metabolic pathway discovery with some power law models that I don't know if you probably know Mike Savageaux that worked here, and he worked on Ph.D. and so that was the mathematical model platform that I was thinking about. That was in 2006 or 7, but I think it can be applied here. And again, this is new, so I'm not sure if it's going to work, but hopefully it will. So, and the goal is try to have a systematic, comprehensive study of this interaction between the host and the microbiome. We just submitted a grant on C. difficile, which is basically known that there are certain patients that can become susceptible after antibiotic treatment. And this is because the antibiotic the treatment disrupt the microbial community. And so we want to understand what happens in these different microbial communities before and after exposure to antibiotic. And we can do it in a quantitative way, semi-quantitative way. We need to tackle this in a more systematic way. And the way we're thinking to do it is illustrated very nicely in a recent review in Nature. This section is in the future in that particular paper, so what to do in the future. And I was struck by it. I mean, it seems like, well, this is something that I can do. I mean, we can do, so let's try to do it. And at least with patch loss, we're trying to do it on some germ-free mice where we can control the initial population of the microflora. So the idea is we do have some time series, longitudinal study. Think about this different microbial community in a single mouse, and we track them over time. And the color here is just an intensity or the frequency or the relative abundance of one community versus another. So it's basically the percentage of this community over time. Now, how do we model this? In this particular review, was suggested to use a kind of a generalized Lotka-Volterra model, you can see here, and which has very nice features. Namely, one is we can interpret these parameters in the growth of the specific bacteria or community, this BI, and this other parameter is the interaction strength between these different species. It could be positive or negative, so they can synergize, so they can exploit each other. Now, again, this is really not mechanistic. We don't know why they do it, but at least we can build a network, and we can, on top of that, we can try to make some inference on, for example, if there are any stable steady state, the system tends to converge, or certain states that are unstable, and how we can shift the population from these different states, perturbing the initial condition or perturbing the system, while it's basically evolving. It sounds doable and easy. There are several computational challenges, though. First of all, we don't have only 4 communities that we track. The species that we track are between 500 and 600 that can be measured simultaneously. And those are called OTUs, operational taxonomy unit, but you can think of that as a species, so just microbes. Now, there is also the problem modeling framework. Yeah, we can use generalized Lotka-Volterra. I also would like to try, the power law formalism. I'm going to show you a little bit how that can be implemented. Well, once we define the model, obviously we need to fit the model, and the choice of the best model is always non-trivial. And with differential equation system, the fitting step is particularly critical sometimes. So this is one, think about this just as a flow of the discovery, knowledge discovery. Basically, these are heat map. Those are real data, though. Those are heat map of the relative abundance of almost 6,000 OTUs, or species, if you like, over time. And you can see here, most of them, the redder they are, the more frequent they are. Most of them, they're not there. So how can we filter and scale down all this massive amount of data so we can actually model it with the dynamical system? Well, the first step is to try to come up with very simple filtering criteria. For example, we can only, or we want to track OTUs that are at least 1 or 2% at any time. So we don't want OTUs that really are below 1 or 2%. And this reduces the size of the the data by 1 log even more. So from 500, we go to 30. Then there is another step we can use to combine OTUs, the species that belong to the same phyla or family, if you like. And also they have the same trend. Those are real data across different mice. So they're fairly consistent. So once we do this, we can start to build a model, forget about the numbers, just the workflow again, and select the model, see the fitting, and there is an interpretation of this parameter that we can use to build the topology of the network. Now, more specifically, this is some data that I worked on like no more than a week ago. We come up and reduce the parameter space from 500 down to 11. And you can see here is basically this OTU or species that is rare that has up to 30, 40% relative abundance. So it comprises many, many different species. The x-axis is time, and we track this mice. This is one single mouse. We track them for 21 days every day. And again, the y-axis is the relative abundance. When you sum them all up, all the species issues sum up to 100%, which usually don't because some species are missed. This is not an exhaustive search. So the model here is a generalized log Volterra. And okay, I'm not sure which model is the right one, right? It could be any combination of the species interacting with each other. Well, let's fit the fully connected model so we don't have to do any model selection. And basically, we assume that each species is connected to all the others. And we could get some decent fitting. Obviously, we are overfitting. We have a lot of parameters that could be neglected. But in general, it worked, at least for this mouse. Now, okay, now what are we going to do? Well, let's look at the values of this estimate, the parameter values. So here, this column will be the alpha, intrinsic growth rate, and this is the matrix, the square matrix of the Bij. Those are the interaction between communities. This is the result of the fitting, one of the fitting. So one thing that I did was, okay, let's try to normalize this and treat this matrix as a correlation matrix. And basically, normalize it between -1 and 1. And see if we can build on top of that the network and the graph. And that's what we did without any constraint. And this is what we get. I don't know if you can see all the connection, but this is basically taking into account all the possible connections with each one with all the others. And obviously, it's not very informative, right? So when we have a correlation, we really don't care about correlation that are very, very small. Like, let's put a threshold. Let's look only at the correlations that are higher than 0.1, for example. And let's see how the network looks. Well, this makes the network much clearer and something that can be analyzed and compare across different mouse. So this is just one mouse, and this is the fitting for this mouse. And you can see that the arches, the branches, the connection have a direction. And also the solid line means it is a positive influence, for example, of OTU 18 on OTU 2. And the dotted line means that the OTU 7 is basically interacting negatively with the OTU 2. I don't know what that means or what that might mean. It can be that they kill each other. So the OTU 7 is killing OTU 2. Or it can be that they compete for the same nutrient. And so OTU 7 is better than OTU 2. So we don't have that information yet, but we will have some information on the metabolomics. So try to see what type of metabolic feature we have at different time, and try to see if there is a shift in metabolism that can explain some of this disappearance on some OTU and one, effect on, try to explain some mechanism about this interaction strand. The other thing that I would like to do on this is basically bypass a little bit this overfitting, because we're really fitting too many parameters and it's really not necessary. But use the information here to seed a power law model. That's the the BST or the S system or the Mike Savageau formalism. So the Lotka Volterra here has a linear and first order interaction. But we're missing, for example, some synergy between 2 and 3 OTUs impacting another one. And that's missed in that particular approximation. Here, we can, again, use this particular filtering and design a model that has a lot less parameter, but is intrinsically nonlinear, and it has some high order effect embedded in the model, formulation. For example, I don't know, just picking one, OTU1, the first component, we can think of it as the production or proliferation of the species. Well, OTU1 has a positive influence by OTU12. And so here we will have the OTU1 that has a growth because the bacteria or the microbe can grow, plus there's going to be another power law, another component OTU12 that is positive impacting OTU1. And here we're going to have OTU7 that is negative, affecting OTU1. It's going to be in this term, which is the degradation. And that's all we're going to put into the model. So a lot less parameters. And so I'm not sure if this is going to work, but at least it's going to eliminate a lot of parameters from the model fitting. The next step is, again, to introduce some knowledge about the metabolism and hopefully some immune system information, correlate some of the dynamics of some cytokines, for example, to the growth or extinction of certain species. Perturb the system and feed the data, similar data, but with antibiotics. So you could see that identify some phenotypes of these communities that they evolve and they shift to certain state that are more susceptible. So what's the difference in the network between a susceptible and non-susceptible community? All these kind of studies that not be done yet, but that's what we would like to do. And I think, yeah, that was the last one. And so definitely, I mean, I need to thank a lot of people, collaborators and mentors, Denise Kirschen and Joanne Flynn for all the data on TB data. And many collaborators here at U of M and members of the lab and some other collaborators that recently appreciated the modeling work, and they used me to basically have an in vivo validation of their hypothesis because they couldn't do it experimentally. And so the only way to do it was, well, let's try with the model. If at least the model, goes in our direction, then we can say something. Well, thank you, and if you have any questions, I'll be happy to answer. Thanks a lot.
Are there, just bringing the two parts of the research together, I mean, are there certain communities that kind of exist along microbiome communities that you see with tuberculosis?
No. I don't think anybody has studied that. To study that? It's a
microbiome, there's a long microbiome.
Oh, absolutely.
There are certain, there's a range of, kind of communities, phenotypes, that's okay.
Yeah, absolutely.
Some of which might exist, in the tuberculosis
Yeah, that's a good idea. The problem I see is that it's hard to get data because you need to, well, where are you going to sample this data? You need to go, that's a problem in TB. The granuloma forms in the lung, and you need to get sample of what's going on at the granuloma level on the tissue, and those samples and those data can, most of the time, can be obtained only sacrificing the host. And so you're missing on a lot in terms of dynamics, evolution of, which we do have in this microbiome, microflora, the gut, because we can actually get fecal samples in this case, and those are going to be, I mean, you can track longitudinally a mouse and the evolution of this. Oh, Bell. Okay.
studying the microbiome.
Okay. Yeah. In the lung. Okay. Yeah, we're working together actually on the U19 with Gary. He's in charge of, well, if we get funded. But I mean, the idea is he's in charge of the immune component and try to correlate shift in the immune response to the microbiome. Yeah, I didn't know. Well, yeah, okay. Well, that's, yeah, absolutely. Thanks. You had a question? You wanted to- Yeah. That's interesting. Yeah, thanks.
How much computer cycles do you need to run some of these big simulations?
Well, so it can be very intensive. If we use a coarse grain model, it's not that computationally costly. We can solve it with 30 minutes. But then we do have, for example, the multiscale where we turn on some receptor ligand dynamics. And recently we can turn on, for example, the lymph node compartment. We can turn on um, the bacteria that we're modeling right now in more detail, and that can take much longer. And there is an issue with the model that is basically replicating 3D phenomena too. This is 2D, so we do have a 3D model. That's what the problem becomes intractable computationally. So lesions, granuloma lesions, typical lesions are between 1 and 3 millimeter. Here we can only capture at most 2 millimeter in 2D. So we need to work on increasing, the size of the grid, and that takes longer, obviously. But also you use a 3D model, and that takes much, much longer. So we're talking about days. And that's particularly relevant if we want to do the sensitivity analysis, because that, we need multiple runs, many, many runs. And a single run in a 3D model can take a couple of weeks. So that's, and we haven't worked on that because we don't have a lot of computer power. Hopefully, more CPUs and clusters, and I don't know, it's coming. So we can use it. Still, yeah. We're using Axiom. We're using Flux now. We have this 500 cores for 2 months, which is it's not going to be enough for research because we're hopefully going to do some research after 2 months. Yeah. Yeah, I think we came up with some numbers. I don't have the exact numbers. Maybe Paul has some numbers, but
Yeah, I know. So, the question is, $7.5 million priority. I mean, it's really something we got to look at.
yeah.
Because it was, by the time we got to where I was, more like 3,000 dollars. And I was just trying to understand, I
Yeah, many grants haven't been funded. I don't know many, but I know a couple that basically say you cannot do that at U of M. I mean, there's no enough infrastructure to do that. And so, I mean, that could be one leverage because, I mean, if we can get money for grants. Yeah, yeah. Two on, 300, yeah. Yeah. What can we do? Okay. Okay.
So in the TB modeling, can you model drugs coming into this and drug susceptibility and see what happens in the system when you put in, say, rifamycin or something like that?
Yeah. Well, we're starting to do that. So speaking to the computational part, the drug is a molecule that diffuses. So the diffusion component in the model is the one that is basically the more computationally costly. So the more component diffusing we have, one will be the drug, the longer it takes to solve the system. But yes, we can use some PK and PD data on certain drugs, especially isoniazid right now, I think we're doing also, right? And we have some unknowns in terms of drug penetration, for example.
Like into the granuloma?
Yeah, into the granuloma, and also maybe in the lung tissue we have some data, but definitely the granuloma is an unknown. But that's the modeling part. We can see and modulate that and see, okay, if we have this penetration, what's going to happen? Can we match at least some data in terms of bug numbers? Because we do know that when you use isoniazid, which is one of the drug, which is never used alone, though it's used in combination. So, but when we use that, we do know that the bug numbers go down. Now the problem is we're killing the weak bugs basically, and so we're left out with the strongest one, and also there is a lot of resistance that is generated during this process. But all these mechanisms are not really known fully, and there are some data in vitro. There's nothing or almost nothing, at least in human or in non-human primate, yet on this susceptibility and bugs. I mean, there are studies. We were collaborating with Pittsburgh and Joanne Flynn on getting some of this data. And yeah, that's one. That's interesting because we can actually use the model and make prediction and give some suggestion on what type of regimen, and also try to make it better. Because the treatment right now is a daily pill that goes on for 6-9 months. So that's not very efficient. There's a lot of people that don't comply.
Yeah, the treatment though right now, as you said, it involves a combination of things, up to like 4 different drugs at the same time. Can you model all 4 of them at the same time, or is this going to be
going to a
$1.5 million computer?
Okay, if you're thinking of the 3D model, well, that's going to be an issue. But in the 2D setting, I think we can do it. Yes, it's just a matter of defining the right parameters, finding the right parameters, and getting more data. I guess we don't have a lot of data on some of the other drugs, at least in vivo. We do have some data in vitro, and we can try to start from there.
And the kind of data you need is like PK data.
Yeah, PK, we don't have, I don't think we have any Ph.D. data on, I know we have something on isoniazid, but that's not in the, I think it's in the rabbit, right? So there's nothing on the non-human primate. So, I mean, those studies have been done partially, but not with the right animal model. And so there is a lot of experimental data that needs to be collected. But in principle, we can do it. Yeah, I mean, it's well, the best guess would be once we have one drug figured out, we can assume some ranges of penetration and diffusion and some key PK that we know in vitro and plug them in and see what happens. We're not there yet, obviously, because many data are not even measurable right now in vivo. So how many bugs are actually there? What's the metabolic state of the bug? So that's important because those drugs, they act on different metabolic state of the bug. So some they kill actively proliferating bugs, some they are not proliferating but they're metabolically active. So we need to refine, what's what we're doing? We need to refine the model to have all these different metabolic states for the bug and then plug in the drug that we know is going to target that specific population or that specific mechanism. So we're working on the bug, to make it better, and with some very basic metabolic transitions, say replicating, slowly replicating, and dormant. And then we're also working on some drug isoniazid, I think, right now. Similar way we can do immunomodulation, and so change. Right now in the model we have tumor necrosis factor. We can do the same thing. We know that if we increase certain molecules, we can do some good to the granuloma. I think the problem is to do it in vivo. I mean, there's no way to do it in vivo right now. And the monkey models are expensive. So before you start that particular experiment, you need to have the money, and you don't even know if it's worth it. I mean, we're talking about $200,000-$300,000 to have a few monkeys and and do the experiment. Is it worth doing it or not? And right now, there are not too many sources for funding, so that's a big obstacle.
Okay. I think we could thank you again. It was really a great talk.
Thank you.
Wide range of quite relevant things that you're doing. So,
All right, thanks. Thank you. Thanks.